talking about cells. Okay, so we noted that our cells have three components to them. One, the plasma membrane. Two, the cytoplasm. And three, the nucleus. But we'll get there, okay? The cytoplasm is inside of the plasma membrane, okay? It is made up of cytosol, organelles, and inclusions. Okay? That cytosol is a very jelly-like substance. Like, it is kind of almost like Jell-O, but a little bit waterier, all right? It has a bunch of cellular elements, components dissolved into it. So a bunch of ions, salt, a whole bunch of other stuff dissolved into that cytosol, okay? It is made up of water, ions, and other enzymes that keep your cell going. Little note for you guys, if you see this little Pac-Man symbol, that's my shorthand for enzyme. Sometimes I will not write out the whole word enzyme. It's that little Pac-Man symbol, okay? All right, we have a bunch of cytoplasmic organelles. We're gonna run through them real quick. The first is the ribosomes. Ribosomes are made of protein and they make proteins, okay? They are the site of protein synthesis, so all of our protein production happens in your ribosomes. They are the assembly line of our manufacturing plant, because basically our cells make a whole bunch of proteins for us to be able to do things. The proteins are what tell your cell what to do, when, and where. Kind of roll with me on that one, okay? Free ribosomes are kind of just hanging out within the cytosol, but you will also have ribosomes attached to the endoplasmic reticulum, which is another one of those organelles that we'll talk about in a minute, okay? Some of those proteins are used to renew the plasma membrane. Some of those proteins are exported outside of the cell. Sometimes they just kind of go wherever they need to be, depending on what the protein is, okay? Next one that we'll talk about is the endoplasmic reticulum. Endoplasmic reticulum. All right, which is a system of envelopes and tubes kind of throughout the cytoplasm, usually pretty close to the nucleus. Okay, you have two types of endoplasmic reticulum in your cells. You have the rough ER and the smooth ER. The rough endoplasmic reticulum just has ribosomes attached right to it, dotted on there. It's named rough because it looks rough. It's just spotted. And the smooth endoplasmic reticulum does not have any ribosomes on it. It's that easy. It's smooth because it looks smooth because it doesn't have any ribosomes attached to it. And the smooth endoplasmic reticulum does quite a bit of synthesizing of like lipids and things of that nature. All right? That's kind of what it looks like. Like I said, quite close to the nucleus usually. And you can see all these little dots. Those are ribosomes attached directly to that endoplasmic reticulum. All right? Next organelle on our list is the Golgi apparatus. You can think of the Golgi apparatus as the Amazon shipping plant of the cell. Essentially, what happens is that products that are produced in the endoplasmic reticulum, whether that be a protein, whether that be a lipid, whatever it is, comes in from the convex, the cis side of the Golgi apparatus, gets packaged in what we call vesicles, and then gets spit out the other side, the concave side. Concave. Hey, I didn't ask you. Okay, Siri, doing too much. And essentially comes in from the cis side, gets packaged up, and then sent out to the trans side. Okay? So that's our packing and shipping guy, Golgi apparatus. And that's kind of what it looks like. And you'll see all these little vesicles that transport things out. Trans side, transports. Trans side, transports. Okay? Hello. This slide kind of gets into the specifics of how that occurs. Read into it. It's pretty interesting. Okay? Next organelle on our list are lysosomes. Lysosomes are so important to your cell. They are the... Oh, this is wrong. I lied. Sorry, guys. I moved stuff. I lied. The mitochondria are the double-walled membranes of your cell that generate most of your cell's energy. You guys all know what the mitochondria is. It is what? Thanks, guys. It is. Mitochondria is the powerhouse of the cell, okay? It is where your cell generates, makes, releases the most energy in the form of ATP. ATP is just a shortened version of adenosine triphosphate. Cells with super high energy demands are going to need more mitochondria, believe it or not. For example, your muscle cells need a lot of energy. If you're running from a bear or something like that, you're going to want your cells to have a ton of energy so that you can outrun the bear and live, you know? So cells that need a lot of energy will have a ton of mitochondria. Cells that don't won't. Mitochondria are a super complex organelle. There's several theories, actually, that believe that mitochondria used to be an entirely different species on its own. It was a bacteria that was living its own life, and it formed a symbiotic relationship or a helpful relationship between our cells and the mitochondria. And so now we have them. We also find that mitochondria has DNA in it that is separate from your nuclear DNA, the DNA that tells all of your cells what to do. Mitochondria also has its own sets of DNA, which kind of alludes to that was a separate entity at one point in time and now became a joined unit. Okay, that DNA is transferred from mother to child. So all of the mitochondrial DNA that you have in your body is from your mother. All right? It is maternally inherited. That's kind of what it looks like. It's that double-walled kind of kidney bean type of dude. It has an outer membrane and an inner membrane. These walls, these wiggly walls, are called cristae, and then the space in between is the matrix. Okay? All right. Next on our list, we have lysosomes. Super important organelle. Our lysosomes contain digestive enzymes that help to break down any waste anything that our cells no longer need, that all gets broken down and sent away by our lysosomes. We also call these our cell's janitorial crew. Question for you though. What would happen if we didn't have lysosomes?
Like if you had internal bleeding, it would just like exacerbate.
Kinda, yeah. On the right track. Any other thoughts as to what could happen without lysosomes in your cells?
Removing the amorphous substance, you're gonna be stuck with a bunch of trash.
Exactly. Without lysosomes, your cells will fill up with a bunch of trash, a bunch of garbage, with a bunch of waste that will eventually become toxic to your cell and kill it. There's only so much waste that a thing can handle before the waste overcomes the good things. When you've had too much stuff and you've kind of been backed up for a few days and you feel sick and yucky, that's what would happen to your cells if you didn't have lysosomes. But like for everything. Think about that. Yeah, think about that. You would just die, straight up. You would be dead. Edema is a different thing. That's more of your lymphatic system kind of holding on to more water, swelling, stuff like that. That edema is a different thing entirely. The next cell, this one often gets confused with lysosomes. They are not the same thing. Peroxisomes break down toxic materials. Okay? So lysosomes are just general waste, like coming to pick up the trash, all that stuff. Peroxisomes are your hazmat suit guys. Hazmat suit guys. All right? They're the ones that are dealing with toxic materials such as free radicals, poisons, alcohol. Your peroxisomes are what's getting rid of all of that stuff. Okay? Where might you find a lot of peroxisomes?
Liver.
The liver. Yeah. You have hella peroxisomes in your liver because you got to get rid of all of the toxins and nonsense that you've been putting into your body. Maybe not. So don't get those confused. Lysosomes, general trash and waste. Peroxisomes, toxic waste. The stuff that really hurts. Bring your hazmat suit. Okay. The cytoskeleton is a network of rods and things that kind of keep and maintain the shape of your cells. Kind of like the bones of your body, but the bones of your cell. Okay? There are 3 different types of rods that we talk about: microfilaments, intermediate filaments, and microtubules. All right. Microfilaments contain actin and a little bit of myosin here and there. Intermediate filaments are just straight protein fibers. And the microtubules are cylindrical structures made up of protein. And kind of shwa. Okay, they're kind of half in, half out. Okay? This didn't move as well as I would have liked it to. But you can see the different filaments, they're different sizes. Microfilaments are the smallest, microtubules are the biggest, and they have different functions. Remember, structure equals function. So their different structures will allude to them having different functions. Okay? Hello. Centrosomes and centrioles. A centrosome is a cylindrical structure kind of in the cytoplasm near the nucleus. And it's comprised of that centrosome matrix and centrioles. And we'll talk about that a little bit further when we get into cell division and what those actually look like. The centrosome and the centrioles are essentially just like spider web arms that help kind of hold and secure the organelles, so they're not just kind of floating up in random space, but kind of keeps everything on almost like a line, almost like a clothesline that you can hang clothes on, but for organelles. Okay. And centrioles act in forming cilia, flagella, and mitotic spindle, and all of these things are used for cellular movement in some way, shape, or form. Okay. Last part of the cytoplasm was the inclusions. Not all cells will have inclusions all the time. Inclusions can be temporary structures. Okay. So they can consist of things like pigments, like melanin, protein crystals, like urea, food stores, all of that jazz. The food store parts, you'll usually see those as lipid droplets or glycosomes. And those are just different ways for your body to store nutrients. Okay. Last major part of the cell is the nucleus. The nucleus is the brains of your cell. Okay? Your nucleus is what provides the DNA that has the instructions for protein synthesis. Remember, there's this process that we talk about, transcription, translation, all of that stuff, that kind of coming up from general bio, stuff like that, transcription, translation, a little bit. I'll go into it a little bit. But essentially, the nucleus holds all of the DNA that gives all of the instructions for making proteins. And proteins are the actionable unit that lets your cells do things. Okay? Your nucleus is comprised of a couple of parts. The nuclear envelope. It's a two-membrane kind of system filled with fluid. Nuclear envelope. Okay, yeah. And then you have nuclear pores. Nuclear pores essentially are an opening in between all of the layers from the outside to the inside. Usually you'll have things like rRNA be formed in there, and that rRNA makes ribosomes, and those ribosomes have to leave your nucleus so that you can make proteins other places in the cell. I know I lost a couple of you there, but that's okay. All right. The nucleolus is that central part of the nucleus that makes those ribosomes. Okay. Subunit assembly. Okay. All right. Your DNA is made up of 4 subunits. Does anybody know any of the names of these subunits? A, T, C, and G. Thymine. You guys all said so many things so quickly. Good job. But you got them. Thymine. Adenine. Cytosine. And guanine. Okay? Adenine binds to thymine. Cytosine binds to guanine. These form molecules called pyrimidines and purines. Okay? your DNA is comprised of those 4 subunits. Okay, I'm going to try to make this as simple as possible. The DNA is packed in your cells with a bunch of stuff, but we're going to go through it kind of layer by layer. So DNA plus proteins form chromatin. All right? Each cluster of DNA and histone protein is called a nucleosome. So you'll start with just straight DNA. Straight DNA is just straight DNA, okay? DNA plus histones is a nucleosome, okay? It'll make more sense when you can see it. All right, let me go from there. Those histones are essentially these little discs. You'll have about 8 of them packed together that wrap around the DNA. And then when they're hanging out on that DNA string with the histones attached, or with the nucleosomes all kind of wrapped together, they kind of look like beads on a string. Kind of like that. But imagine that each one of these like little knobs, it has a bunch of DNA wrapped around it. Okay? Each histone will wrap some DNA around it. Okay? So then when the chromatin is open and we are ready to do like transcription, translation, we call that extended chromatin. Okay? That's that active region where DNA is being copied and made into other copies. Condensed chromatin has tightly coiled nucleosomes and is the inactive form of DNA. Chromosomes are going to be your highest level of organization in chromatin and also the most condensed. Humans have humans have 46 chromosomes. Humans have 46 chromosomes, point blank period, give or take 1 or 2 here or there, okay? So it goes like this. We have DNA plus histones, which is a nucleosome. And we have extended, condensed. Extended is active. Condensed is clearly not. I'm not done yet. Don't take the picture yet. Eventually, that condensed form, once they're condensed enough, will form chromatin. Okay? That chromatin will condense even further and become a chromatid. And then 2 chromatids will cross and become your chromosome. So your chromosome, now you can take the picture, is just very highly dense chromatin wrapped all up. Okay? That's all a chromosome is. But all of that is just DNA. It's just wrapped up DNA. Okay? It looks like this. This is kind of what I was showing you there. You start with your DNA, you get the histones wrap it up, have nucleosomes, those nucleosomes get even more condensed, you get chromatin, that chromatin condenses even further and becomes a chromatid, becomes an arm, Once that chromatid joins with another chromatid, you have a chromosome. Okay? Don't worry about it. No stress. No fuss. Kind of talk about is interphase. Interphase is kind of broken down into 3 steps. Okay. Wait. 3 steps. First of those is the G phase, or the growth phase. Growth 1, gap 1 phase. Okay? That is the very first part of interphase. Interphase is a very metabolically active time in the cell's life. This is where the cell is doing all kinds of growth. It is getting bigger. It's making more organelles. It's doing everything it can to be a big, strong, tough dude. Okay? So this is get swole time. All right? This is your bulk. Phase. This is essentially where all of your DNA starts to replicate itself. Because at some point in time, coming down the pike, this one cell is going to split into 2 cells. So in order for that to have the correct number of, correct amount of DNA, the correct amount of anything, this is the point in the cell's life where it is synthesizing another copy of all of that genetic material so that it can eventually pull it apart and cause it to be in 2 different cells. Okay? And then the last phase that we talk about is the growth 2 phase, G2. And that's where the kind of centrioles finish copying themselves. Any enzymes that are needed to finish kind of cell division happen. We know that our chromatin is extended in that S phase because that is the active form of chromatin. Okay, we have the active form, the extended, and the condensed chromatin is inactive. Condensed chromatin, inactive. Okay. During all 3 of these partial interphase phases, the cell is still carrying out all of its normal activities. So it's still, taking in nutrients, it's still producing waste, it's still making more proteins. It's just doing all of this stuff in addition to its normal everyday functions. Okay? That's kind of what it looks like, very generally. So this whole big part of it is interphase. The majority of your cell cycle is interphase. And then there's the teeny tiny little bit of it that is the mitotic phase, which is where we make our secondary daughter cell and split. Okay? So you got your growth phase, and there's checkpoints all throughout this process, because if at some point in time your cell determines that something's not right, something didn't get replicated properly, and it passes through that checkpoint, it's not going to pass through that checkpoint. Your cells are going to say, hey, this is wrong, eat it, get it out of here, I don't want it, it's going to make something bad. Okay? So those checkpoints are put in place so that you don't get like an overdevelopment of a wrong type of DNA, cell, whatever. Okay? And then when we talk about the mitotic phase, that's the part we're talking about, the actual cell division that's going to be going on. Okay? So cell division or mitosis, is just division of the nucleus. Okay? And all of that happens after interphase. So after it's grown, after it's grown, synthesized, and then grown again, then we'll get into cell division and dividing that cell and its nucleus. Okay? So we have the process of mitosis, and then later down the line we have cytokinesis, which is the actual separation of those 2 cells. So mitosis has technically 4 phases to it, but cytokinesis is the unofficial 5th. Does anyone remember any of the names of the mitotic phases?
Prophase.
I heard one. There's a prophase. I heard a what bit of huh? Anaphase. I heard it over here. I heard a metaphase. What's that last one? Telophase. Yeah. And then our unofficial final, cytokinesis. Okay. So, going with our little PMAT. We have prophase. Prophase is the largest, longest stage of mitosis. Lots of stuff happens in prophase. Okay. In early prophase, the chromatin condenses down into chromosomes. Remember how we were talking about in the synthetic phase, our chromatin is extended? It has to come back down to a more condensed phase because it's no longer actively being copied in what we call transcription. It is now just hanging out. It needs to be small enough that it can be pulled apart. Okay? So our chromatin condenses into chromosomes. Those chromosomes are made up of two threads called chromatids. They're usually sister chromatids, right? That's that kind of X-like thing that we look at when we're talking about DNA. One is a chromatid, one is a chromatid. The chromatids are held together by a centriole. I lied. They're held together by a centromere. Okay, that's that yellow guy in the center there. And then eventually, the centriole that is hanging out near the nucleus, because remember, that's just kind of the arms that help to keep all of our organelles in place, right? The centriole separates from itself. The mitotic spindle forms, and we're off to the races, okay? Then in late prophase, I'm just going to abbreviate that as late pro. These centrioles continue moving away from each other, and the nuclear membrane begins to break down, right? So we have that nucleus that's hanging out within our cell, and that nucleus has layers to it. It has the envelope, it has the membrane, it has all that stuff. It starts to kind of break apart. Why might it need to break apart? Why do we need the nucleus to break open? Don't tell me, tell them.
So you can get the genetic information out?
So that we can actually get to the genetic information. If it's all wrapped up in the nucleus, I can't do anything about it. I can't get to it. Can you? No. You can't eat a candy with the wrapper still on. I mean, you can, but you want to be that guy that's eating paper? I don't. So you have to break open that nucleus so that you can get to the good stuff on the inside. Okay? So this image is just kind of showing you what these phases look like. Some of the slides that we look at in lab have some of the phases of mitosis, I believe. So you can take a look at that if you want to sometime next week. But interphase is basically just the cell growing, doing a bunch of stuff. All of your DNA is pretty loosey-goosey and ready to be copied, right? Because of that synthetic phase. Early prophase, it starts to condense down again. You start getting the centrioles moving, starting to get some nuclear breakdown. Late prophase, that nuclear envelope is almost completely kind of broken down. Okay? Next phase of mitosis is metaphase. Metaphase, the middle phase, middle, right? Makes sense. Is kind of where all of your chromosomes line up along the middle of the cell. And these centromeres are lined up along the equator. So if this is my cell and I have a whole bunch of DNA, all of my chromosomes have hung out here, and my centromeres line right on up. Okay? Anaphase, just the shortest stage of mitosis. This is where the centromere splits. So this thing that was holding them together is no longer. Okay? Also in metaphase, you kind of get the mitotic spindles starting to reach towards their chromatid that they're trying to get to so that they can prepare for the next part. Just pulling them apart. Okay? And then the last official phase of mitosis. Oh, sorry, there's an image. That's what it looks like. It's actually really cool to see cells doing this IRL because it is something that you can watch. Like there actually might even be a video at the end of this one, but you can see the cells going through each phase through. Well, you can't really see all of prophase going on, but you can definitely see metaphase. You can see it kind of just pull itself apart. It's really cool. Cool. Okay. So telophase is when chromosomal movement kind of stops. The chromosomes end up on opposite ends. Let's make that a little bigger. Your sister chromatids will end up on opposite ends of the cell after getting pulled by that mitotic spindle. It starts to kind of re-envelop in a nuclear and a new nuclear envelope on both sides. And then during cytokinesis, the cell completely splits into two guys doing their own thing called daughter cells. Daughter cells. Okay? So telophase is when all the chromosomes stop moving, the nuclear envelope starts to reform, all of the like cell stuff starts to happen, and then cytokinesis is where it actually fully breaks apart. Let's break down that word cytokinesis. Oops. Cyto. Cell. Kinesis. Movement. Cytokinesis. Okay? Ah, there is. This is not as fun as the IRL one, but it's still pretty cool. If it wants to play. It doesn't, but that's okay. You guys can play it later. Or Google it. That's what Google is for. Google mitosis, cell splitting, and you can watch a cell split, IRL. Well, online IRL, in front of your eyes. Okay? Cool. Sick. All right.